Anna Strampelli. Anna, here, can you share your screen? Great, thank you. Anna is a third-year PhD student in the Crisanti Lab at the Imperial College of London. Her research focuses on developing self-limiting genetic population suppression strategies in the main malaria mosquitoes, Anopheles gambice. I hand it to you, Anna.
Hi everyone, can you hear me and see my screen?
Yes.
Perfect. Okay, thank you very much for having me today. The title of my talk is A Male-Drive Female Sterile, or MDFS, Strategy as a Population Suppression Tool for the main malaria mosquito, Anopheles gambice. So genetic strategies for population suppression constitute novel additions to the malaria control toolkit, and they aim to reduce the disease burden by reducing the number of mosquitoes in the wild that can transmit the parasite. They present multiple desirable characteristics. I've listed 3 of them here. First of all, they're very equitable in their effects because they provide area-wide protection, which is independent of factors such as age, wealth, or gender. They're very targeted and species-specific because they rely on sexual reproduction for their spread, and also they're cost-effective. So genetic strategies tend to target regions of the genome that are essential for female reproduction or viability. And this is because for mosquitoes and other insect species, it is the number of females rather than that of males that determines population size. And one such region is the intron 4 exon 5 boundary of the double-sex gene. So you can see in this picture taken from Hammond and Kiro 2018. So double-sex is a gene involved in sex determination in mosquitoes and other insect species. And it has a male-specific transcript that you can see on the top, as well as a female-specific transcript on the bottom. And specifically, the female-specific transcript has an exon 5, that is not present in the male isoform. And as you can see in the shaded region, the intron 4 exon 5 boundary has been found to be very highly conserved across species of the Anopheles genus, indicating an important evolutionary role. And it has been targeted using CRISPR-Cas9 in previous genetic population suppression strategies. And what was found is that disruption of this female-specific exon 5 in both double-sex alleles causes complete sterility in Anopheles gambice females, and also their inability to take a blood meal. Heterozygous females for the disruption, and all males are fertile. So the male-drive female strategy, female sterile strategy, is what I've been working on as part of my PhD, and it consists of a transgene that is depicted here in blue, carried by males, that when present in heterozygosity is transmitted to 100% of the progeny rather than the Mendelian 50%. So we say that it drives in the progeny. At the same time, females inheriting this transgene are completely sterile. And so it's a male-drive, female sterile approach. And males are fertile, and they can continue transmitting this allele to the future generations. And eventually we reach population suppression because of the reduction in fertile females. Now, whilst female sterility is what we want to achieve with this strategy, it also means that at each generation, in terms of allele spread, 50% of the progeny, the female progeny, is a dead end because they do not transmit the gene to the next generation. And so a strategy that has been proposed to potentiate the MDFS approach is to couple it to what we term a sex distorter. So a sex distorter is shown here as a red allele. And what it does, as the name suggests, is to bias the sex ratio in the progeny towards males. So rather than having a 50-50 female to male ratio in the progeny, we now have 95% males and 5% females. So note that the sex distorter does not change the activity of the MDFS allele. It still drives in the progeny and is inherited at 100% frequencies. The females inheriting it are still sterile. The only difference is that these females now represent a much smaller proportion of the progeny, 5% as opposed to 50%. So we have a bigger proportion, 95% of males now that don't bite and don't transmit the disease, that can, however, pass on this gene to future generations, potentiating the spread of the MDFS. Another thing to note is that the sex distorter is a non-driving allele, so it's inherited at 50% frequencies through Mendelian laws. And modeling on both the MDFS alone and the MDFS coupled to the sex distorter suggests that these are both viable strategies for population suppression if they were to be released in the wild. So we went on to design an MDFS strategy in Anopheles gambice by targeting the female-specific exon 5 in our wild-type colony in our lab So we injected wild-type embryos with the construct that you can see here on the right that codes for the Cas9 under a VASA2 promoter, a GAT-RNA specific targeting the female-specific exon under the ubiquitous U6 promoter. And it's not shown here, but also an actin CFP fluorescent marker to visualize our transgenics. And at the bottom of the slide, you can see the kind of MDFS colony that we now have in the lab. And it's heterozygous for our transgene at the double-sex female-specific exon 5. And the reason why we thought that this construct could confer the MDFS phenotype that I described, so male drive, female sterile, is due to the dual activity or expression pattern of the promoter that we used, VASA2, to drive expression of the Cas9. So on the male side first, we know that VASA2 is a germline specific promoter and specifically acts in the early germline in the premeiotic diploid cells. And so following Cas9 cleavage in the male germline of the double-sex wild-type allele, this allele is repaired primarily through homology-directed repair, which is a repair mechanism that uses the homologous chromosome as a repair template. And so what effectively happens is that our transgene following cleavage is copied into the cleaved chromosome. And so whilst our males are heterozygotes for the MDFS allele is effectively inherited by all the gametes through this homing process that occurs in the germline, and so therefore drives in the progeny of males with the MDFS construct. So at the same time, VASA2 also exhibits what we call somatic leakiness, in the sense that its expression is not specific to the germline, but it also occurs across somatic cells in the body of females. And so because of this activity, Cas9 will also cleave to some extent alleles in the somatic cells of this female, so double-sex alleles, which will then be repaired this time, because we're outside of the germline, through a process called non-homologous end joining. So this is an error-prone mechanism that typically inserts indels at the cleaved sequence and will disrupt the coding sequence of the gene. So again, whilst our females are heterozygotes for our MDFS construct, they're actually homozygotes for a null allele at double sex at the phenotypic exon, because one of the alleles is disrupted by our transgene and the other one is disrupted by the indel caused by Cas9 cleavage. And so with many of these somatic cells being disrupted, we expected our females to be sterile. And so we conducted a phenotypic assay, which I'll describe in a second, to assess the main aspects that would affect the efficacy of our strategy. On the male drive side, we looked at the fertility of our MDFS males, as well as the fertility of MDFS males, which also contain a sex distorter. And we also looked at the inheritance rate, which we called homing rate, of the MDFS construct in their progenies to see if it derived. And on the female side, we looked to see at the penetrance of our MDFS sterility phenotype, so how sterile our females would be. So we set up three cages: our wild-type control cage that contained 50 females and 50 males, our MDFS experimental cage, which contains 50 females and 50 MDFS males, and our MDFS and sex distorter cage, which contained 50 females and 50 trans-heterozygote MDFS sex distorter males. Following mating and blood feeding, we allowed these females to individually lay in separate cups, and then we counted the egg and larval cup output from all of these different cups. And for the MDFS, as well as the MDFS with the sex distorter, because we're also looking at the inheritance of our constructs, we screen for the relevant fluorescent markers, which in MDFS were CFP, and in the other line, MDFS sex distorter, were CFP and RFP, with the RFP being associated to the sex distorter. And results in this front were very encouraging. So what we first found was that male fertility did not vary across the three different lines. So we looked at two different parameters, as I said, egg output and larval progeny. And on top, you can see the MDFS males also containing the sex distorter. In the middle in blue, we have the MDFS males, and at the bottom, we have the wild-type controls. Each dot represents the output from one cup in which a female that was mated to one of these males was laid. And as you can see, there's no significant difference in any of the parameters looked. In addition to this, another encouraging result was that the MDFS allele drove in the progeny of these males. So both in the MDFS and sex distorter males, so on the top, and in the MDFS males on the bottom, we found that their progeny inherited the CFP fluorescent marker associated to the MDFS allele at frequencies between 99.5 and 99.7%, which is much higher than the expected Mendelian 50%. So it successfully drove in the progeny. And in fact, the sex distorter allele, which was a non-driving allele, as I mentioned earlier, was inherited at 50% frequencies, as was expected. The last thing that we did was to look at the fertility of our MDFS females. So we took between 14 and 15 cups per line. And when the larvae became pupae, we sexed them. And then at adulthood, we tested the fertility of the resulting females. So for the MDFS, we tested over 1,000 females. For the MDFS and sex distorter, because there was this large male bias, we could only retrieve about 15 MDFS sex distorter trans heterozygote females. And we tested them against 50 wild-type female controls. And again, what we found was very encouraging. So firstly, we saw that all pupae coming from the MDFS crosses looked male-like. So on the left, we have a normal wild-type male pupa, which has this characteristic spike in its genital area, which is missing on the right in the female wild-type pupa. And on the center, we have our MDFS pupa that, as you can see, looks indistinguishable from males. And as adults, our MDFS females displayed the intersex phenotype I had described before, when females have two null alleles at the double sex X and 5. So on the left, we have a wild-type male with characteristic plumose antennae and also claspers facing the abdomen. On the right, we have a wild-type female with no plumose antennae and no claspers. And in the middle, we have our intersex, which has some plumose antennae and a clasper that is actually inverted towards the back. So none of these females that we found, so over 1,000, were able to blood feed or laid any eggs. So they were completely sterile, as in contrast to the wild-type controls that we tested. So the inability of females to bite means that they're also unable to be transmission vectors of the parasite, which is actually really important. And the data that we have on both the male drive, so the fertility of the males and the high homing rate in the progeny, and on the female sterile side, so this complete sterility that we see, is very encouraging in terms of the success that we can predict for these strategies. So both the MDFS and sex distorter strategies as population suppression approaches in Anopheles gambiae. So next steps in my experiments include an analysis of the cleavage frequency at the double sex locus in MDFS individuals, because I'm very curious to know what percentage of the wild type alleles has been cleaved. I'd like to dissect intersex females to further characterize the phenotype. And also we're planning a cage trial to further assess the potential of these strategies as population suppression approaches, at least at the beginning in a caged setting. Thank you very much. Thank you to my supervisors, Barbara Fasolo and Andrea Crisanti, and to everyone else in the lab, and to you all for listening.
Thank you very much, Anna. Great presentation. So now we're open for questions in the Q&A chat. I see that there was one question that I think it was already clear by your presentation, that says the inability to take a blood meal. Do you mean that they can feed, or they are not interested in feeding on blood?
They try to feed, but they can't. There's so you can definitely see that they're trying. They're attracted. They can smell the blood. They can try to feed. They try to feed, but they're not able to take blood
in. Thank you. Thank you for your insight. I just have one query. We're witnessing Anopheles funestus colonizing new herbal habits as opposed to vegetatively. Oh, I think this was from.
This was for Leon.
Yeah, this was from the previous talk. Interesting study. Can you predict if in the wild a sterile male will be selected?
A sterile male. So if you mean if, well, I don't think that will be the case because we're targeting a female-specific exon. So the male transcript is unchanged. So I don't think that would happen from.
I think perhaps the question is maybe also, are they likely to be competitive? You know, these males that you release, are they likely to be able to compete with the wild type males?
Right. So we haven't done the study yet. In terms of fertility, we know that there's no fertility differences between our males and wild type males. And because again, we have we're targeting a female-specific site, I don't see how that would happen, but it's not something we've looked at or studied yet.
Okay, well, I wanted to jump in with a question as I get to cheat as a panelist. So I wanted just your overall idea. Like we've been hearing a lot about these different genetic technologies to control mosquitoes and these different gene drive technologies. Where exactly does this fit in? Where would this be used compared to the other types of technologies? For example, the 2018 gene drive targeting double sex. How exactly is this going to fit in control in Africa?
So in terms of, in general, genetic population strategies, as opposed to other types of control measures, they're definitely seen as complementary tools and not just standalone tools. In terms of the various types of genetic population suppression strategies, as you've said, there's, well, the two main categories: self-sustaining ones, such as the gene drive that you described, and self-limiting ones like the one I described, which is more, as the name suggests, contained in its use. I think that both of them will have, so again, they'll be complementary to each other. There's cases in which, this MDFS strategy is kind of seen as a strategy that would work with repeated releases of frequencies of about 25% compared to the total population. And so in regions where the population of mosquitoes is, in which this can be achieved with the population size of the mosquitoes, this could work. There's other regions in Africa where releases of 25% of our males compared to the total amount would be impossible, and there maybe a full-on gene drive would be more suitable. And also the self-limiting strategy that I've described here. So I didn't have time in my presentation to go into the differences between self-limiting and self-sustaining, but the self-limiting strategy can also be used not just as a standalone or as a population suppression strategy, but also as an intermediary product in the development pathway towards self-sustaining versions, because those are yet to be approved. So it can also be a useful intermediary product in their approval pathway.